This is ongoing global and regional adaptive evolution of SARS-CoV-2, presented by me, Nash Rochman, with co-authors Yuri Wolf, Guilhem Farr, Pascal Mutz, Feng Zhang, and Eugene Koonin. This work is currently available at PNAS. Please note, the analysis I will present here was conducted at the beginning of 2021. While the features emphasized remain relevant now, in July, recent mutations are not reflected. Unprecedented sequencing efforts have produced literally millions of genomes of severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, the virus responsible for COVID-19. Understanding the trends in SARS-CoV-2 evolution is paramount to control the pandemic, but analysis of this enormous dataset is a major challenge. The construction of a single high-quality tree using any of the existing advanced methods is computationally prohibitive. An iterative construction may seem an obvious solution, such that a global topology is constructed based on a subset of sequences available at an earlier date, and later sequences are then added into the existing tree. However, this approach induces artifacts through the inheritance of deep topologies which differ substantially from any maximum likelihood solution corresponding to the complete alignment. Therefore, building on the available techniques, we utilized a divide-and-conquer approach, which is not subject to these artifacts and may additionally be employed for datasets which cannot be structured by sequencing date, including metagenomic analysis. This approach leverages two ideas. First, a diverse representative subset of sequences may be used to establish the deep topology or tree skeleton, corresponding to any maximum likelihood solution over any alignment. Second, uncommon substitutions relative to consensus are unlikely to support deep branches in an unrooted tree. Leveraging this methodology, we were able to arrive at a robust global topology with 8 major partitions and 3 emergent variant clades. The most prominent variant clade, V2, corresponds to B117. Others, including notably B1351, correspond to dramatic regional signatures but were not yet topologically informative. Using this tree and the associated metadata, we showed that the ongoing evolution of SARS-CoV-2 over the course of the pandemic is characterized primarily by purifying selection, but a small set of sites appear to evolve under positive selection. The receptor binding domain of the spike protein and the nuclear localization signaling associated region of the nucleocapsid protein are enriched with positively selected mutations. The mutational signatures of each phylogenetic partition are largely composed of sites within these regions, and these mutations form a strongly connected network of apparent epistatic interactions. The first mutation in this set to emerge was spike 614G. An ensemble of likely NLS-associated mutations followed, rising to global prominence summer 2020. Evolution of beta coronaviruses with high case fatality rates, including SARS-CoV-2, was accompanied by accumulation of positive charges in the N protein that are thought to enhance its transport to the nucleus. There are 7 prominent N mutations in the vicinity of known NLS signaling motifs or other regions responsible for nuclear shuttling. Conceivably, these substitutions, some of which increase positive charge, might contribute to nuclear localization. This highly unusual cluster of multiple signature and positively selected mutations in the N protein is a strong candidate for experimental study that could illuminate the evolution of SARS-CoV-2 pathogenicity. Following the rise of these likely NLS-associated mutations, in fall 2020, an ensemble of mutations in the spike protein became dominant, and yet another wave of emergent variants bearing conserved mutational signatures. While viral diversity within each geographic region has been steadily growing for the entirety of the pandemic, assessing regional diversity reveals 4 distinct periods. First, there was a period of rapid diversification into region-specific phylogenies ending February 2020. This was followed by a major extinction event and global homogenization, concomitant with the spread of D614G in the spike protein ending March 2020. NLS-associated variants across multiple partitions rose to global prominence March to July during a period of stasis in terms of interregional diversity. Finally, beginning July 2020, multiple mutations, some of which have since been demonstrated to enable antibody evasion, began to emerge associated with ongoing regional diversification. These trends may be indicative of speciation. And with that, I'll thank you for listening. Thanks again to my co-authors and to support from the Intramural Research Program of the NIH, and thanks as well to ISCB and the COVID-19 subgroup for the opportunity to present.